Step outside and take a look around your environment. With a little observation, you will likely see many organisms that look and behave differently, but you are just one person, with a single experience in that single moment. Think about if every person did this at the same time around the world and wrote down every organism they saw and put it together in a single list. This would not only be insanely large, it would also have a lot of redundancy, as people in similar locations and biomes would see similar organisms. In order to study organisms properly, scientists need a way to classify all of the vast diversity within organisms that we see on our planet. And once an organism is classified and we know how it relates to other organisms, a lot more work can be done to study other aspects of that specific species. In this video, we will talk about how scientists classify organisms and how, at times, the process can be a little tricky. Scientists classify organisms into specific groups called taxa. These groups start at a level that is very broad and gradually get more specific based on traits that organisms share. We can see the names of these groups here, which starts with the most broad category of a domain and then gets more specific with kingdom, phylum, class, order, family, genus, and species. The example shown here is for the red fox, which ends with the species name of Vulpes and the binomial name of Vulpes vulpes. Recall in video A3.1 you learned about binomial nomenclature, which is that two-part scientific name generated from this hierarchy. We can see another example here for our human species, from domain down to species. You can see that we share the first four broad taxa with the red fox, but diverge at the order level. While placing organisms into taxa groups like this can be helpful, there is a major downfall in the sense that the groups are created arbitrarily and can cause disagreements between scientists as to which ranks should be separate. Is this group different enough to have a separate genus? Should the group of organisms be placed into the same family? What happens when there is a speciation event and the organisms begin to diverge from one another? At what point in time should a group be changed? These are all questions that speak to the difficulty of using this system. But lucky for us, we have a way out of this with a different system that we will look at on the next slide. To remedy the issues brought about by the taxonomy hierarchy, scientists have another way to classify organisms, which is based on evolutionary relationships. The basic idea here is that if organisms have evolved from a common ancestor, that is the evidence required to place them into a taxonomic group. That way we can look at a specific group and say these organisms share traits because they have inherited them from their common ancestor. This is then a very powerful tool which can allow scientists to then make predictions that can be helpful to many fields. One example of this can be seen with the discovery and development of aspirin. For centuries, diverse cultures utilize willow tree bark, genus Salix, to treat pain and fever, but it wasn't until scientists applied predictive cladistics to the broader Salicaceae family that the full chemical potential of this group was realized. By identifying salicin as a defensive compound evolved in a common ancestor to protect against herbivores, researchers were able to predict the presence in closely related branches of the evolutionary tree. This guided them towards meadowsweet, Filipendula ulmaria, and various poplar species, which contained high concentrations of the compound. This predictive capacity of evolutionary relationships allowed chemists to isolate and eventually synthesize acetyl salicylic acid, transforming a shared botanical trait into one of the most widely used life-saving drugs in human history, aspirin, which I am sure many of you have used or will use at some point. The process of classifying organisms through cladistics involves putting organisms into groups based on DNA sequence divergence along with morphological characteristics which deduce the common ancestor that species shared. A group of organisms that originated from a common ancestor is called a clade, and we can put clades together into a diagram called a cladogram. A very basic image of a cladogram can look like this. It has groups of species of organisms at the end, branching points called nodes that reveal a split from a common ancestor, and an implied arbitrary concept of time in the past as you move away from the species at the end. So if I were to ask you, which organism did A share a more recent common ancestor with, you would say B, as A and B have the same node in common that is more recent than this one. If I were to ask at what point did organisms A, B, and C all share a common ancestor, you would say this point. The points, or nodes, are important here because they dictate the branching point between two different clades. Each individual line at the end of the diagram, whether it represents an individual species or a group that is not broken down further in the cladogram, is its own clade. Looking at it that way, we can highlight three different clades as seen here. In addition to that, each node makes a clade that includes every organism that comes after it, and because of this, organisms can occupy more than one clade. So as an example, if we take a look at the most distant common ancestor between these three organisms, it would be here, which means that this common ancestor is the node that identifies this clade that has all three organisms. But then organisms A and B share a more recent common ancestor, and thus they can be identified in a separate, smaller clade like this. And of course, we have the individual clades to add to that we showed before. All clades must include all organisms that relate to the shared common ancestor. So if you were to try and say that this is a clade on the exam, it would be incorrect, because organism B also stems from the common ancestor at this node and therefore has to be included. Compared to what we saw on the last slide of the hierarchy of taxa, if we were to look at human classification based on the clades we are in, it would look something like this. We can say that two organisms shared a common ancestor based on their DNA similarities and differences. While this is helpful, it is also helpful to try and piece together when speciation events happened in the past. And the cool thing is, we can use information like DNA evidence to get an estimate of how long ago organisms diverged from a common ancestor. To make sense of this, we need to understand that differences in DNA sequences between organisms are generally caused by mutations. And if we assume that mutations occur at a constant rate, which we have a lot of evidence for based on living organisms today, we can use DNA as a tool to go back in time, which is a method we call the molecular clock. This of course comes with a grain of salt because mutation rates are affected by the length of generational time, the size of a population, and the intensity of selective pressures and other factors. In any case, it does provide some insight as to when speciation and other events could have played out. When looking at the genetic divergence between humans and chimps, who are about 1.2% different, and using a mutation rate of 1 mutation per billion base pairs per year for mammals, which is a bit on the slower end, we can deduce that chimps and humans diverged from a recent common ancestor around 6 million years ago. We can do the same thing for chimps and bonobos, calculating they have an average genetic distance of approximately 0.4%. This would put the common ancestor around 2 million years ago. Again, this is on the slow end of the estimation scale. This is a powerful tool that biologists can use to talk about evolution and classification, as they are constantly seeking to understand all of these relationships between all living things on Earth. Cladograms are constructed using genetic information, which can come directly from base sequences of genes, or it could come from amino acid sequences of proteins that are derived from the base sequences of genes. In any case, using genetic information is the best tool we have to properly classify organisms into clades. There are billions and billions of base pairs to go through when an organism's DNA is sequenced. We rely on computer computation within specific programs to do the heavy lifting in the sequencing aspect, so we can then use the data to compare and apply it to different organisms for classification. Let's go through an example of how we can use technology to make this comparison and also create a cladogram, or phylogenetic tree, with this information. To do this, we're going to use two programs, which are the NCBI site, which stands for the National Center for Biotechnology Information, and the Clustal Omega site, which will run a comparison of all the DNA sequences we acquire. We will be comparing three species: Panthera leo, the lion; Panthera pardus, the leopard; and Panthera tigris, the tiger. And the gene we will focus on is the mitochondrial cytochrome B gene. It is one of the most effective and widely used DNA sequences for species identification and phylogenetic analysis. This 1,140 base pair protein coding gene is highly valuable because mitochondrial DNA has a high copy number compared to nuclear DNA, making it easier to amplify even from degraded samples. Let's start with using the programs. I will walk you through each step, and I suggest simply watching first and then doing it later on your own to practice the process. Alright everybody, so you have two links on your screen for these two websites that I just talked about. I'm going to go ahead and show you how you can use these to kind of take a look at DNA and genetic information and create a cladogram from it, comparing a few different species. So on this first website, the NCBI website, I'm going to go up here and I'm going to type in the specific gene that we're looking for, which is cytochrome C, and we're going to do this first for Panthera leo, which is the lion. I'm going to hit enter, and we can see the cytochrome C right here for the lion. I'm going to go ahead and click on this specific gene. I'm going to scroll down and go to the legacy gene page. Click on that, and then from there, I'm going to scroll down as well, and I'm going to go to this FASTA link, which basically just takes the gene and puts it into text form. So I'm going to click on that, and we can see we have the genetic code for this specific gene. So what I'm going to do is I'm going to go ahead and highlight this entire thing, including all of this extra information at the top, and I'm going to go ahead and copy it, Control C. I'm going to go over to my other program, the Cluster Omega, with the link that I had you go to, and I have it on protein type, and I'm going to go ahead and paste this in here. I guess I should probably switch this to DNA. And what I'm going to do, I know it says Panthera leo right here, but part of this kind of gets cut off when it gets compared. So I'm just going to take this, I'm going to copy this, and I'm going to paste it right here so we know exactly the name of the organism at the front. And then I'm going to go down to the very end of this. I'm going to hit enter, and I'm just going to do the same thing for the other two organisms. So we just did this for the lion. Now I'm going to do this. I'm just going to go back here, click this top part right here to get back to the home page. And I'm going to do the same thing for the leopard, which is Panthera pardus, and for the tiger, which is Panthera tigris. So I'm just going to speed this process up, but it's the same process. We're going to copy and paste it into the Cluster Omega the same way. Okay, so now as we can see, I have all the information pasted into this program. We have the first gene here, the entire sequence, followed by the second one for the panther, and then we have the third one here for the tiger. So I just have all of that together, and I'm just going to go down here. You can give it a name if you want, and I'm going to go ahead and click Submit. And it's going to go ahead and process and read through all of this information. And of course, it kicked out an error here. Two sequences can't share. Let's see. Oh, I think the problem is I put spaces here. I should put an underscore there. So for the tiger, and let me go up to this one, add in an underscore, and then this one as well. This should hopefully fix it. Let's go ahead and try again. I'm going to scroll down and hit submit. Okay, it looks like it's running now. And it's going to go through and it's going to kick out a comparison. So we can see here, we have the tiger, the lion, and the panther. And it takes all of the DNA sequences and it puts them next to each other. It aligns them all next to each other. And when there are these stars below each individual star or asterisk here, it signifies that all the specific bases in that position are the same. So we have the same basis if there is an asterisk star there. So you can see like right here, we have a C base here for the tiger, a C for the panther, and a T here for the lion. So it doesn't have a star there, so we have a difference. So we can kind of scroll down and just take a look at some of the differences here between the DNA sequences with these three organisms. And we kind of scroll all the way down. It compares all of the bases together. Now what we can also do in this program is we can click on the guide tree, and when we click on this, is it takes all this information. You can see right here, you can make it a little bit bigger. So it takes all this information and actually puts it into a nice little cladogram that we have here. And this is showing us that we have the panther and the lion that are sharing a more recent common ancestor than with the tiger. And that's just based off of this individual single gene alone, right? This is the only thing we inputted, but just based off of this alone and the evolutionary history based on the genetic differences, this is what the program can tell us. So I recommend going and trying to do this on your own, maybe find a different gene, look at some different organisms, and you can try it on your own. At this point, you know some of the terminology around how cladograms are structured. But let's go over the details again and add in a few new terms. First off, the shape of a cladogram can be drawn in a few ways. You can keep the lines at 90-degree angles like this, or you can have it look more branched at a different angle. There is no right or wrong way to do it. It's just your preference. The end of each line is called a terminal branch. and the stem of the entire cladogram, which represents the distant common ancestor to all of the terminal branches, is called the root. Each branching point is called a node, which is generally when one line splits into two groups, though in some cases it can split into three or more than three. The goal of the cladogram, as you know, is to provide a framework for the evolutionary past and show connections between organisms or groups of organisms. But this is all an assumption based on the current data we have available. As information is always changing by the addition of new data points, cladograms will change as well. So you should not look at a cladogram and assume that all of the relationships are true, but instead look at it and think, based on the information we have today, this is how we think evolutionary history connects the organisms that are present in the diagram. Let's take a second look at a real scenario in which cladistics, classification, and new information can be used to help make sense of evolutionary relationships between species. Angiosperm is a term that describes all flowering plants, which is a very large clade, or phylum, that has hundreds of families classified under it. One of those families is Scrophulariaceae, which has a common name of the figwort family. For a long period of time, as botanists were classifying flowering plants, this specific family was a dumping ground for plants that shared similar flower structure. This led to there being over 5,000 or so species that stemmed from this family based on morphology. But over the last 30 years or so, with huge advancements in DNA sequencing technology, it was discovered that based on the DNA of the species within this family, many of them did not share recent common ancestry with one another. With this new genetic information, the old figwort family had over half of its original 5,000 species reclassified into other families that showed a proper clade with common ancestry. While this was a very large reclassification event, as many species were moved around the same time frame, new DNA evidence is constantly being used to place organisms into proper clades based on common ancestry. We have already looked at the specific name groups that make up the hierarchy of taxa, which include domain, kingdom, phylum, class, order, family, genus, and species. But it was not always this way, specifically when it comes to the largest and first groups that are used to classify all living organisms, the domains. The traditional classification system split organisms based on cell type, which are prokaryotes versus eukaryotes. But in 1977, microbiologist Carl Woese proposed a new domain system after studying ribosomal RNA which, based on those studies, split prokaryotic cells into two separate groups. Based on these genetic structures, three distinct domains were created: bacteria and archaea, which are prokaryotic, and eukarya, which consists of all eukaryotic celled organisms. This was an important shift because it revealed that archaea, despite looking like bacteria under a microscope, are genetically and biochemically as different from bacteria as they are from humans. This three-domain system replaced older models, emphasizing that the most fundamental split in life isn't between plants and animals or prokaryotic and eukaryotic cells, but between the three ancient lineages defined by their unique genetic machinery.